Gene libraries, genomic and cDNA libraries.
Introduction. What is DNA library? A DNA library is a collection of clones of DNA designed so that there is a high probability of finding any particular piece of the source DNA in the collection. There are two types of DNA libraries, which are genomic library and cDNA library. The genomic library contains DNA fragments representing entire genome of an organism. Meanwhile, the cDNA library contains only complementary DNA molecules synthesized from the molecules in a cell. What is genomic library? A genomic library is a collection of bacteria chromosomes which have been genetically engineered to hold the entire DNA of an organism. Besides that, a genomic library is a collection of genes or DNA sequences created using molecular cloning. Genomic libraries can be constructed using various hosts like plasmids, bacteriophage, lambdas, cosmids, yaks and many more. What is cDNA library? A cDNA library is a combination of cloned cDNA, which also known as complementary DNA, fragments inserted into a collection of host cells. cDNA is produced from fully transcribed mRNA found in the nucleus and therefore contains only the expressed genes of an organism. In eukaryotic cells, the mature mRNA is already spliced, hence the cDNA produced lacks introns, and can be readily expressed in a bacterial cell. While information in cDNA libraries is a powerful and useful tool, since gene products are easily identified, the libraries lack information about enhancers, introns, and other regulatory elements found in the genomic DNA library. The diagram at the left side showed the genomic DNA library and the right side showed the cDNA library. The table below showed the differences between genomic and cDNA library. Next, construction of cDNA libraries. First, isolation of selected mRNA specific from cells/tissues. Then, purification of mRNA from total RNA after reverse transcription to produce first strand of cDNA. Next, synthesis of complementary strand cDNA to form double-stranded cDNA. Continued by ligation of linkers or adapters to both ends of synthesized, double-stranded cDNA. Next, digestion of restriction site by restriction enzymes. Then, cloning of cDNA into suitable vectors. Last, transformation of vector cDNA into a suitable host, form cDNA libraries.
Next, purification of mRNA from total RNA. First, high-quality mRNA selection required both effective removal of rRNA and non-mRNA species. Then, mRNA isolated from animal cells contained long stretches of polyadenylate acid tail. After that, oligo-DT matrix chromatography, commonly used to purify mRNA. The polyatail will bind complementary to the T residues attached to the surface of the matrix. Then, selected mRNA is eluted out with low salt buffer. Reverse transcription of mRNA to cDNA is through first and second strand synthesis. At first strand synthesis, initially, add oligo-DT and dNTP into the sample. Oligo-DT will bind complementary to the polyatail. Then, reverse transcriptase synthesis, the first strand, making use the mRNA as template, an mRNA-DNA hybrid is formed. At second strand synthesis, first, terminal transferase at specific sequence of nucleotide to the 3' of first strand cDNA. Then, poly-cDNA primer can be synthesized complement to the poly-G tail added at the 3' Next, DNA polymerase can synthesized second cDNA strand by adding dNTP, complementary to the first strand. Linkers is a small fragments of blunt end nucleotide sequences. It ligate with cDNA by T4 ligase, and contained restriction sites. Restriction enzyme cut at the restriction site, forming sticky end and then ligation of cDNA with the vector. Meanwhile, adapters has the small fragments of sticky and oligonucleotide sequence. It is also ligated to the cDNA by T4 ligase. But cDNA can be incorporated into the vector. The diagram at the left showed the processes of linkers, and at the right showed the processes of adapters. There are different selection of cloning vector. If less than 10 kbp, plasmid vector will be selected. If around 10, 15 kbp, bacteriophage vector will be selected. If around 45 kbp, plasmid vector will be selected. If around 300 kbp, bacteria artificial chromosome will be selected. If around 1000 kbp, yeast artificial chromosome will be selected. Besides sizes, cost and techniques involved will be taken into consideration. Next is the construction of genomic library. First, partial digestion of chromosome. Next, cloning of partial fragments into vectors. Then, transformation of vector into host cell. Last, screening of positive clones. The diagram showed the details of all steps involved in construction of genomic library of a phage. Step 1, isolated genome. Step 2, cutting isolated DNA into suitable sized fragments, using restriction enzymes. Step 3, vector with a DNA fragment. Step 4, introduction to a suitable host, multiplication, screening, identification and characterization of clones. Last step, maintenance of set of clones.
Next is the determining titer of library. Titer of the library can be determined after a genomic library is constructed with the viral vector, so that to approximate know how many infectious viral particles were successfully created in the library. Transfected cultures of E. coli of known concentrations, and plated on agar plates and incubated overnight. Then, counted the number of viral plaques. Most viral vectors carry a marker that allows clones containing an insert to be distinguished from those that do not have an insert. This will able to determine the percentage of infectious viral particles that actually carrying a fragment of the library. A similar method can be used to titer genomic libraries made with non-viral vectors. Titer of library is initiated with transformed E. coli and spread on agar plates and incubated overnight. The titer of the transformation is determined by counting the number of colonies present on the plates. Next is screening library. One of the key elements required to identify a gene during cloning is a probe. Probe is a piece of DNA or RNA used to detect specific nucleic acid sequences by hybridization, which also mean binding of two nucleic acid chains by base pairing. Probe is radioactively labeled so that the hybridized nucleic acid can be identified by autoradiography. There are two types of probes, which are homologous and heterologous probe. Homologous probe is a probe that is exactly complementary to the nucleic acid sequence that are searching. Heterologous probe is a probe that is similar to, but not exactly complementary to the nucleic acid sequence that are searching. Next, I will talk about screening a cDNA or genomic library. First, immobilize members of the library onto a nylon membrane and denature them. Next, prepare a redealable probe and denature it to make it single-stranded. After that, hybridize the probe to the library of clones and wash the excess probe and expose an X-ray film. Last, isolate the positive clone and analyze. There are alternative ways for screening cDNA. By using a DNA probe with a homologous sequence. By using an oligonucleotide probe based on a known amino acid sequence. By using an antibody against the protein of interest. And plus/minus, or differential screening.
Next is the applications and uses. It is used to express eukaryotic genes in prokaryotes. cDNA does not have introns, and therefore can be expressed in prokaryotic cells. It is useful in reverse genetics where the additional genomic information is of less use. It is used for subsequently isolating the gene that codes for the mRNA. It is used in discovery of novel genes. It is used to obtain the sequences of genes for analysis, amplification, cloning, and expression. Besides that, it is used for analysis through bioinformatics. The complete cDNA library of an organism gives the total of the protein as it can possibly express. cDNA sequence gives the entire relationship between organisms through the similarity of their cDNA. It is used for in vitro study of gene function. It is used for reproducing eukaryotic genomes as the amount of information is reduced to remove the large numbers of non-coding regions from the library. It is also used as a probe for DNA microarrays or as a clonal inserts into bacteria chromosomes. It is used in commercial production of proteins and other biological products. It is useful in carcinogenic identification. It is used in study of alternate splicing.